Okay, so we'll start a new chapter today. Okay, so let's start the class. So we are going to discuss a chapter called as Strategies for Enhancement in Food Production. Now, whenever we talk about strategies, exactly what is the meaning of strategies? Strategies mean ki? Plans, right? And why are we talking about improvement of plants? Just to improve the quality of food, right? So food boli kole obviously all over the world, no? Because population is high, food demand to basi, food demand to jetia basi, so we need to improve the quality of the food, the quantity of the food, etc., etc. So we are going to discuss certain things regarding this in this chapter. What exactly we are going to study over here is yate a contents ki ase, yate ase strategies, kenuka dhoroner strategies, jeneke animal husbandry, right? Next we have something called as plant breeding. The third would be tissue culture and the fourth is single cell protein. These four are the contents of this chapter. As far as animal husbandry is concerned, this is something that would be dealt in your zoology class. So we'll be starting our class with plant breeding, okay. So plant breeding, tissue culture, single cell protein, this is the content, basic contents 9. So ekhiniye pohim. So before writing the notes, plant breeding, what we understand by the word plant breeding mane ki? Ekdam simple, you don't go by definition. Plant breeding mane ki? What you do? You take two plants, right? What you do? Cross. Hybridization is a word that we understand. Cross kora. Is a cross to korilu, kiho karne korilu? So that what? You get a desired result, right? The offspring juntu banao, by which amar mote juntu offspring lage, just to have that offspring, what we do? We select two best parents, we make a cross and then we produce the offspring. And that process is called as what? Plant breeding. What exactly we have done? We have changed the traits of that plant. Adult plant or genetic makeup to ami change korim, right? By cross korim, first time cross korim, second time cross korim, then you will keep on crossing the plants, hybridization kori thakim, and ultimately we will try to change the gene and ultimately enuka edal plant produce korim jontu niki amar mote it is the most efficient plant and it has economic value. Next, after the process of plant breeding, how do you go for plant breeding? What are the steps of plant breeding? We'll have to discuss. Plant breeding korar pisot, the offspring that we get, juntu offspring ba juntu notun parent ami form korilo, you have to check whether that is desired or whether that is the best or not, right? Adult plant talk, ami best boli ketia kom. Dudal plant loiso, dudal plant cross koriso, cross kori pela offspring eta paiso. That is the whole process is called as plant breeding. Product of plant breeding, juntu ami plant palu, the offspring palu by F1 generation, by F2 generation, juntu ami palu, hetu best boli ami ketia kom. When it will have certain characteristics like what? Maybe it will have high yield, right? Production basic kore, or it will be disease resistant, right? Bemar semar ba puksuke bhalke na ki khabo nware, so disease resistant, high yield, and third could be ki resistance against ki adverse environmental conditions, right? It could tolerate drought, frost, etc. So ebila qualities thaka edal plant ami jodi banao, that process is called as ki plant breeding. As far as the steps of plant breeding is concerned, the first step is something called as collection of the germplasm. Right? Ami ki koru? Germplasm mane ki? The collection of the genes. Adult plant, jot gozi ase, wherever it is grown, maybe it's in India, maybe it's in your own place, maybe something outside the country, etc., etc. A single plant or jiman gene ase, hey gute genes ba, gute plant or collection to ami koru, that is what is called as germplasm. Right? So you collect that germplasm and then from the germplasm, the next thing what you do is you select. Prothomte collect korilu beleg jegar pora tasot ami select korim ki select korim the parents that we are going to make a cross with parents ki to select koribo laibo no so select korilo that is the first step first ki select collection second is selection select korar pisot we'll go for the cross after the cross ki form koribo product the third step would testing the product to test koribo laibo desired mote hoise ne nai so that is the third one and ultimately we have the last step that is testing and commercialization. If it is everything is all right, then we can obviously release it to the market, right? So these are the 4 steps how you will go for plant breeding. So exactly what is plant breeding? It is the changing of a trait of a plant. Why do you do plant breeding? To get a desired plant. What is a desired plant? A desired plant might be with characteristics like high yield, disease resistance and resistance to adverse environmental conditions, right? So, heikini bostu ami discuss korilo. Next is, in this chapter we have to talk something called as green revolution also. Yeah, green revolution kotha poribo laibo, because whenever we talk about food, whenever we talk about increase in food, we always have to talk about green revolution. What is green revolution? Is nothing but the increase in the food, right? Eta process by eta start korisilo jar karone niki eta duta particular plant ami increase koraisilo quantity, and which two plants are we talking about? We are talking about rice and wheat, right? Green revolution on ki hoisile, it is a revolution which has increased the quantity of food. Which food buli kole to rice or wheat, so that those two are the two crops, right? So green revolution part to poribo laibo, father of green revolution, Indian father of green revolution, aru kuntu plant ami choose korisilo, rice or kuntu plant, wheat or kuntu plant. We have something called as a dwarf variety, heblak ami janibo laibo, what was the gene that was present in that dwarf variety, ekini poribo laibo. So ekini hol up tomar about green revolution. After that we have single cell protein. What is single cell protein? Single cell that is used as a food, that is what is called as a single cell protein. Not necessarily rice, wheat, ebilake khabo laibo, kisman microorganisms also could be taken in as food, right? And this all are included in single cell protein. And the last part would be tissue culture. Tissue culture, we have certain words called as callus, micropropagation, etc. etc. Aru eta word is explant. Ekhiniye hol lump sum. But if you are going to write the note, obviously we have to write a long note, right? So start kora plant breeding buli. Strategies for enhancement of food. No, the question is food enhance kiyo koribo lage? Why you have to increase the amount of food? The basic answer or the most common answer is because of the growing population, right? Enhancement kiyo karne lage? Population growth ho karne. So we'll start with the note. Strategies for enhancement, total it line likha. So with these methods, what we can do is we can go for enhancement with food production, and if food or amount of food increases, then starvation no hobo, no, everyone will get food. It is not a very complicated chapter, nor does it requires a lot of explanation. Note to poriba, note to porile, you'll understand. हाँ লিখি লোৱা কোনটো ইম্পৰ্টেণ্ট ফ্ৰম এক্সামিনেশ্যন পইণ্ট অফ ভিউ মই মাৰ্ক কৰি দিম এক সেকেন্ড হ্যাঁ কোনটো পয়েন্ট which one regarding first point মানে art and science of Okay, art and science of changing the traits of plants in order to produce desired characteristics. Did you get it? Okay. Saccharum barberi and Saccharum officinarum, OFFICINARUM. Two alag species. Genus, genus same है लेकिन species अलग अलग है. Right? Okay, so if you are talking about enhancement, we have to use certain techniques. If you are talking about techniques, the first technique that we are talking about is plant breeding. What is plant breeding? Making a cross between suitable parents, that is what is called as cross breeding, and obtaining a desired character. What you do in the process? You change the trait of a plant, and when we talk about a trait, what we understand? Something to do with the genes. Right, something to do with the character of a plant, that is what is called as a trait, no? Our adult plant or character ami keneke change korim, tar ami kiba kibi koribo laibo na, gene modification koribo laibo. Yeah, so changing the trait to produce a desired character is called as plant breeding, right? Plant breeding techniques use kori pelai duta specific crop or bahut besi enhancement hoisile, and which two crops are we talking about? Rice and wheat. Actually the proper way of writing is wheat and rice. Rice and wheat, exactly, it's the same meaning. Kintu mainly kam kihor upor kora hasil, wheater upor first or types of rice upor. So that is what is called as green revolution. Some examples in which the improvement was seen, as far as these examples are concerned, no, iman basic question regarding these rice ahanai. So you don't have to by heart this portion. As far as this is concerned, Sonalika, Kalyan Sonia, এই দুটাৰ ওপৰত question আহিছে. Question to কেনেকে আহে যে যদি suppose Sonalika দি দিব and the question will be asked, this is a variety of rice ne, wheat ne, rye ne, sugarcane ne ki. So you have to know that these are the varieties of wheat. So this one is important, this one is important. Next, talking about sugarcane, the example has been given so that just to understand that in the plant, in this plant also plant breeding has been done, right? We have given two examples, one that grows in northern India and the other that grows in southern India. As we have already discussed, no, to go for the process of plant breeding, we need to choose the parents first, right? Aru ami plant breeding ki ho karne koru kiba eta bhal bostu pabole. So what they have done is, since this plant had certain drawbacks, right, and the North Indian people also wanted to have or grow sugarcane with good yield, good quality. So what they have done, they have crossed these two plants and obtained a new plant. As far as the name of the new plant is concerned, he naam to diya nai kintu. It's a Saccharum species, right? Parents kitar naam ase, kintu species ba offspring to naam to nai. So we'll know that both these varieties of sugarcane has been crossed to make a better variety. A better variety keneke korim? Obviously plant breeding, right? Hebor. You have to change the traits. Gene mutations koribo, right? DNA study koribo. Or maybe you can go over crossing and crossing, right? Okal first generations after generations you be cross koriba. Tetia holeo kisuman modifications dekha pam. Okay? Sugarcane to buza nai, no? So, A2 example kiho karone disu, just to show that in sugarcane also plant breeding practice has been done. Sugarcane or plant breeding kiho karone korise, kiba eta bhal sugarcane pabole, right? Bhal sugarcane keneke paba, duta sugarcane cross kori pelai, duta parent cross kori pelai, ami notun variety pam, right? First to bhal sugarcane kiyo lage? Why do you want? These people, no, the people that were growing in northern India, so protect according to the soil that we have, no, India jiman ba anywhere in the world, no, depending on the soil culture or depending on the soil quality also, plants vary, right? So jekate eketa crop grow korile quality to ekei nohoy to, right? So ki hoy North India to sugarcane hoisile, South India to sugarcane hoisile, North India A2 variety hoisile, South India A2 variety hoisile. What they have seen is A2 variety is much better as compared to this variety. So what did these people want? They wanted to grow this plant in their own land. কিন্তু তাহাঁতে এইটো বস্তু নি পেলাই নৰ্থ ইণ্ডিয়াত ৰুলে হ'ব জানো নহ'ব কাৰণ চইলটো বেলেগ না চইল বেলেগ ক্লাইমেট বেলেগ ৰাইট চ' হোৱাট দে হেভ ডান ইজ তাহাঁতে দুটাই ক্ৰছ কৰিলে ক্ৰছ কৰি পেলাই প্ৰডাক্ট ফৰ্ম কৰিলে সেই প্ৰডাক্টটো গ্ৰ' কৰিলে এণ্ড দেট ইজ অফ আ ডিজায়াৰ্ড কোৱালিটি আণ্ডাৰষ্টুড So this is just an introduction of plant breeding. What is plant breeding? Nothing but a cross. Why do you go for plant breeding? To obtain a desired product, right? There are certain plants where plant breeding has been done and we have written certain examples. Next, the desired product should have certain characteristics. What are the characteristics that we expect from a plant or from a good plant is crop yield thakibo lagibo, disease resistance thakibo lagibo, or tolerance thakibo lagibo. Tolerance against what? Tolerance against adverse environmental conditions or any other conditions. Releasing and commercialization. So plant breeding korile, what we do first? We first collect the germplasm. What is germplasm? We are talking about the entire collection of genes that you find in that particular crop, right? That is what is called as germplasm. Next you test the parents, next you test the superior recombinant. Superior recombinant mane after the cross, the product that we have obtained, that is what is called as recombinant. You have to test the recombinant also whether it is desired or not, right? And once everything is all right, then you go for commercialization or releasing it a market. Now talking about germplasm, germplasm mane ki collection of genes. Kihor genes? Eta particular crop or gene, eta particular crop jote jote grow kore. It's not only confined to a particular area. Wherever the crop grows, you have to collect the crop from each and every part of the world, right? So that you get a entire collection, and that entire collection will be called as ki germplasm. So heidal plant jote jote grow kore sobor pra collect koribo lagibo. Collect korile ki hobo? You have an idea, or you have all the information about that plant, right? As far as the germplasm is concerned, collect kopra koriba. Next point likha. So collect kopra koriba either within the country or from other countries. You have to remember two words, indigenous or exotic. Especially what is exotic? What is the meaning of exotic? Tar upor question ahe. Something that is brought from outside, no? India bahirot. So the first step is you need to collect the germplasm. From where will you collect? Either it could be indigenous, it could be exotic, right? If you are going for collection, the collected portion that you have collected, no, generally it is maintained at low temperature, right? And in what form will you collect? Yeah, you have to collect the genes, but in what form? You cannot be going to a plant, take the gene and come back. So what are you going to bring? You are going to bring the propagules. What is a propagule? A part. I think you have heard something called as vegetative propagation, right? Propagation mane ki? Reproduction, right? Vegetative propagation, corpora reproduce small parts of. So generally in small parts, and that is what is called as propagules. Now, whenever we are talking about collection, not necessary in each case it is possible to collect the propagule. Sometimes, especially in case of fruit trees or ami ki collect koribo lagibo, generally the plant is collected. So these two lines tells us about what? The process of collection. Either you go for the collection of a propagule or a small part, or sometimes the plant itself and you grow it in a particular area, right? So fruit trees or keneke tumi kuntu part kati ahiba, fruit tu kati aniba ne plant dal kati aniba. Fruit tu lagibo, you have to talk about the gene, you have to see the quality, because in the fruit plant, as far as plant breeding is concerned, economic importance basic your fruit tour hai, right? So in that case ki kore, you go for collection of that plant also, baby plant ani pelai at a particular area gozai diba and you maintain that area, that is your germplasm. From there you do the experiment. Okman cells lo microscope otolot saba modify koriba, etc. But for the case of study, you need to collect it. Our collection, propagules most common, but sometimes also in the form of trees. Clear? Are we at the line too? Okay, we understand. Right. So this is the first step. After the collection time, next is you have to select the parents. Gigi collect kori anisa no, tar best to lobo lagibo. You have to choose the parent also. So number 2 likhim. After collection of germplasm, the next is evaluation of the parent, or you have to select the parent. How will you select the parent from the collection? By the process of hybridization. Buji paisa? Jimanpur collect korila, tarpa kunto best parent keneke gom papa. Tar offspring to sai pelai, right? So by the process of hybridization, what you do is you evaluate. As far as অকল hybridization বুলি নহয়, তাৰ আৰু কিছুমান words আছে আমি লিখিম একেনি. Next, pure lines are created. Pure lines জানুনে self pollinated plant এডাল cross কৰি পেলাই যোনটো offspring produce কৰে তাক আমি কি বুলি কম? Pure line, right? It's not, the word is not referred to cross pollinated plants. Pure line ki edal plant single parent tak cross kori pelai aru eta offspring produce korise, that's a pure line. Cross hole ki hobo, two different parents mixture hoi jabo no. Okay. Kuntu etu collection of germplasm is needed to solving problems like, okay, collection of germplasm is needed to solving problems like loss of germplasm due to expansion. So plant breeding korute ami first step ki korisu, collection of germplasm. Germplasm mane ki the genes, car genes, he plant dalor genes, he genes to tumi ko pra collect korisa, belek belek jagar pra collect korisa, collect kyo koribo lage, jot ase to asei. বুঝি পাইছা নে প্ল্যান্ট ডালটো য'ত আছে তাতে আছে if suppose a plant is growing there, let it grow there, why should I go there and collect it? কিয় কালেক্ট কৰিব লাগে, কিবা এটা থাকিব ন কালেক্ট কৰা, কালেক্ট কিয় কৰোঁ জানি নেকি সেইটো যদি বাই চান্স কিবা কাৰণে ফেক্টৰ্ছৰ কাৰণে নষ্ট হৈ যায়, নষ্ট হ'লে যে প্ল্যান্টটো হেৰাই যাব ন. So to save that, what are the problems? Problems like loss of germplasm due to expansion of agriculture. Agricultural field বিলাক জেগা বঢ়াই বঢ়াই গৈ আছে, যেতিয়া বঢ়াই যাব সেই নতুন জেগাখিনিত অলৰেডি প্লান্টছ আছিলে, ৰাইট, নাইকিয়া হৈ যাব. হিউমেন এক্টিভিটিজ, কিছুমান ফিল্ডছত ঘৰ বনাই দিলোঁ, হয়. পিঅ'ৰ লাইন মই লিখাই দিম, অলপ পিছত লিখাই দিম, কিন্তু একদম চিম্পল মিনিং হ'ল পিঅ'ৰ লাইন কি হয়, আ টাৰ্ম দ্যাট ইজ ইউজ ফৰ সেল্ফ পলিনেটেড প্লান্ট. সেল্ফ পলিনেটেড প্লান্ট এডালৰ অফস্প্ৰিংটোক আমি কি বুলি ক'ম, পিঅ'ৰ লাইন. আকৌ সেই F1 জেনেৰেশ্যনটো আকৌ ক্ৰছ কৰিম, আকৌ কি পাম, F2. অকল এটা ছিংগল পেৰেণ্টৰ পৰা যদি ডিজায়াৰ্ড অফস্প্ৰিংছবোৰ পাই আছোঁ, দ্যাট ইজ হোৱাট ইজ কল্ড এজ পিঅ'ৰ লাইন. দুডাল প্লান্ট যদি ক্ৰছ কৰা তেতিয়াহ'লে সেই ক্ৰছটো বা সেই F1 জেনেৰেশ্যনটোক আমি পিঅ'ৰ লাইন বুলি নকওঁ. Pure line hobo lagile, it should be pure. To be pure, it should be of a single parent. By the word pure, what we understand, gene exchange hobo nalagibo, karon dudal plant cross korile ki hobo, duidal plantor gene ahi pelai offspring to form koribo. Yes or no? Understood? Okay. Second pointer under it etu. Right? When we are talking about selecting the parents, how will you select? By the process of hybridization. Hybridization mane ki you make a cross between two parents. Hey duta parent to select koribo lagibo, right? Hey process to keneke koru ami, first ami parent gita select koru, right? Select keneke koru, there are various methods, we'll talk about it. Select kora pisot we make a cross, right? Desired due to F1 generation but desired product, that product could be it might need mutation breeding also, so that to change the gene, F1 generation or gene change koribo bisarisa, if you want to change the genetic makeup, what you will go for, you will do mutation. I think you have heard something called as induced mutation. Mutations could be natural, it could be induced also, right? Natural mutation to natural, right? Man-made jodi mutation hoy tetal tak ami ki boli kom induced mutation, and we have a word called as a mutagene. Mutagen ba mutagene ji kwa certain types of chemicals, certain type of substances which you use to produce a mutant or mutation, right? So plant dal select korilo, tar cross korilo, tar mutation breeding korilo, so that we can change the gene, right? Next is sometimes we go for polyploid breeding also. Polyploid breeding means again something related with the genetic makeup itself, haploid, diploid, polyploid. I think we understand. Haploid ki one set of chromosome, Normally organisms will have 2 sets of chromosomes, so we are diploid, right? Sometimes what happens is for economic importance, we go for polyploid breeding also, we increase the sets of chromosomes. Suppose if a plant has 2 sets of chromosomes, maybe the size of an apple is this much. You want to change the size of an apple, you want to make it big. So, that we ki koribo libo, obviously gene change koribo libo, so we can change the gene, we can increase the chromosome, etc., etc. So, talking about increasing the chromosome, a word that is called as polyploid. So, by these, ami ki korim, ultimately we will produce the recombinant. Clear? Select korilu, first collect korilu, collect kori pelai parent select keneke korim, eblak korim, cross korim duta parent or mazot, offspring produce korim. Offspring to ki buli kom, recombinant buli kom, right? So, these are the steps that we are going to do. As far as selection is concerned, selection of whom? Selection of the parents. From where will you select? Select it from the germplasm. Select keneke koriba. Dekhi bhal lagile hetu select koribo paru. Cross koriyo tar offspring to bhal lagile hetu select koribo paru. Right? So, first ami selection to likhasun. লিখি লোৱা This part of a note talks about what? How will you do the selection, right? So, selection first koribo labo, selection of what? Selection of the parents. So, how will you go for selection? These are things that we do for selection. Either you go for mass selection or pure line or clonal selection. So, mass selection, as the name suggests, juntu bhal dekhilu hetu uthai loi anilo. We are talking about morphology. Right, we are talking about the phenotypic characters, right? Adult plant ase, plant dal dekhat dhunia, morphology bhal, that is what is called as phenotypic characters. Based on phenotypic characters, if you have selected, that selection will be called as a mass selection. Or the next method of selection is you can make a cross and then, or you can make pure line, right? Pure line bonai pelai select koribi, you will select amongst the pure line. What is the pure line? You will take a self-pollinating plant, you will allow it to fertilize. Dar baby to ami grow korim, hey baby to jodi bhal, hey to ami select korim. Buji paisa na nai, what is pure line? Yata question ki ahe, iman details to question ahe pure line kar karne kora, mass selection kar karne kora. Mass selection generally cross pollinated plants or karne koru, self pollinating plants or karne ami we go by the method of pure line selection. So we are talking about selecting the parents. How will you select the parents? There are three methods of selecting. First, you talk about the phenotypic characters, you talk about the morphology, juntu bhal hetu uthai loi anilo. Second, you make a pure line and then you select. How do you make a pure line? The pure line word itself talks about which type of a plant, self-pollinated plant. A plant will self-pollinate, produce its babies, hey baby to grow korila, hey grow kora baby to jodi bhal, you will collect it. That is what is called as a pure line, right? Or else you can go for clonal selection. Clonal ma likhi aso. I think you have heard the word clone. Clone boli kole, which type of reproduction is it associated with? Asexual reproduction. So you produce clones and you select amongst the clones, that is what is called as clonal. Select duta buzi palu, either morphology or selected from a pure line or selected from a clone. Clone kenege produce koriba, adult plant loba, asexually reproduce koribol diba, the offsprings would be called as what? Whenever we talk about asexual reproduction, no, tar jimanbur offsprings produce kore, all of them are same, yes or no? Yes, so they could be called as what? Clones. So you select amongst the clones and that is what is called as clonal. These are the three methods by which you can select the parent. Pure line boli koi karon tat kuno planter mixture na thake no. Dudal plant jodi beleg cross kora na, cross fertilization, self fertilization. Self fertilization or product to hodai pure. Karon edale plant involved, tar baby to plant dalor nisinai hobo. Right? But if you are talking about cross pollination, tetia hale ki hobo? Dudal beleg plant cross koribo, dudal beleg plant thakile, we'll have a mixture of two different genes coming from the two different plants and the two different parents. Right? Hekarne pure buli koise, karon it is something that is done in self-pollinating plants.
Mass selection mainly for cross-pollinated
মাছ সিলেকশ্যনটো কি বুজি পোৱা নাই মাছ সিলেকশ্যন যেতিয়া কৰে নহয় জাষ্ট এটা জেগালৈ গ'লা গৈ পেলাই যোনডাল ভাল বস্তু দেখা পালা সেইটোৱে উঠাই লৈ আনিলা ৰাইট আৰু ইন নেচাৰ সবতছে বেছিকৈ কি হয় ক্ৰছ পলিনেশ্যনে হয় চ' আমি মোৰ ফ্ৰিকুৱেণ্টলি আমি কি দেখা পাম ক্ৰছ পলিনেটেড প্লাণ্টছৰ অফস্প্ৰিং দেখা পাম ৰাইট চ' সেই অফস্প্ৰিংটো দেখাত ধুনীয়া বা মৰ্ফলজি গুড সেইকাৰণে উঠাই লৈ আনিলোঁ দেট ইজ হোৱাট ইজ কল্ড এজ মাছ চিলেকশ্যন বেছড অন জাষ্ট মৰ্ফলজি আমি তাৰ ভিতৰৰ কেৰেক্টাৰ্ছ একো এনালাইজ কৰা নাই ৰাইট আৰু ইন জেনেৰেল কোনবিলাক প্লাণ্টছ বেছিকৈ থাকিব অভিয়াছলি ক্ৰছ পলিনেটেড বেছিকৈ থাকিব ৰাইট চ' দেট ইজ হোৱাট ইজ কল্ড এজ মাছ চিলেকশ্যন আৰু পিয়'ৰ লাইনটোত আমি স্পেচিফিক প্লাণ্ট ল'ম কোনটো প্লাণ্ট ল'ম চেল্ফ পলিনেটেড হ'বই লাগিব তেতিয়াহে আমি পিয়'ৰ লাইন প্ৰডিউচ কৰিম আৰু পিয়'ৰ লাইনৰ ভিতৰত আমি চিলেক্ট কৰিম ৰাইট এণ্ড দ্য থাৰ্ড ওৱান ইজ ক্ল'নেল চিলেকশ্যন আমি কাৰ মাজত কৰিম যোনে নেকি ভেজিটেবলি তোমাৰ কি হ'ব ৰিপ্ৰডিউচ কৰিব and whenever we talk about vegetative reproduction the offspring that they produce obviously look alike and they could be said as clones also so you select it from the clones and that is what is called as clonal selection or eta example kufri safed potato thik tanake mass selection or eta example wheat or eta example kalyan sona variety to ami keneke obtain korisu by making pure line right একেই থাকিব নহয় একো নাথাকে তাৰ ভিতৰত তুমি গোটেইকেইটা গ্ৰ' নকৰা ন যিকোনো এটা উঠাই লৈ আনা বুজি পাইছা আৰু এনেকুৱা হ'ব পাৰে যে no doubt we are talking about a collection of clones out of the entire lot of clones no কাবাৰ ডেভেলপমেণ্ট আৰ্লি হ'ব পাৰে কাবাৰ ডেভেলপমেণ্ট অকণমান শ্ল' হ'ব পাৰে ৰিগাৰ্ডিং I mean depending upon the environmental characters and other factors also genetic makeup-টো একেই কিন্তু গ্ৰ'থটোতো অকণমান বেলেগো হ'ব পাৰে ন হয় নহয় ৰাইট The next one is hybridization. EE सफ़ेद पोटैटो কি উল্টা পুল্টা লাগিছে কোৱা সবৰে ক্লিয়াৰ হৈছে নাই নে কিবা ডাউট আছে কোৱা
তাতে কালেকশন অফ জার্মপ্লাজম এতে এখন কালেকশন মানে এটু যে লিখছে কালেকশন অফ জার্মপ্লাজম এটু বুঝিয়েছো তারপর ইভালুয়েশন এন্ড মানে সিলেকশন অফ
সেইটোৰ আন্ডাৰত সেইখিনি how will you select as far as selection is concerned you can go for any method that we have described there যিকোনো মেথড এটা এপ্লাই কৰি পেলাই তুমি প্যাৰেণ্টক সিলেক্ট কৰিব পাৰিবা বুজি পাইছা এটা সিধা সিধি লাগিল ঠিক আছে ৰাইট এনেকুৱা নহয় যে এটা প্যাৰেণ্ট সিলেক্ট কৰিবলৈ গুটেইকিটা মেথড ফলো কৰিব লাগিব not necessary you can go for any method for selection right এতিয়া hybridization point to soa aru soa whether you have understood or not what is hybridization making a cross between whom cross between two genetically different parents right cross between two different parents ami kihor karone koru to get a desired character that desired plant that we have made no by making a cross hey plant tot car characteristics thakibo of single parent and both parents so this is the example that we have given a example tot ki koise je we are combining two plants eta plant with high protein আৰু এটা plant with disease resistance right দুইডাল plant cross কৰি পেলাই যোনটো offspring produce কৰিবা হে offspring টোত কি থাকিব it will be best protein quantity ও বেছি থাকিব plus তাৰ disease resistant হ'ব so that would be a desired plant that we wanted আৰু desired plant ৰ কাৰণেটো আমি plant breeding কৰিছো right so for the process of plant breeding we can go for hybridization also for hybridization you do this clear So for plant breeding, how will you go for plant breeding? You can go for hybridization also. You can do hybridization. Plant breeding keneke korim? Plant breeding hybridization or through koribo parim, right? Hybridization ki hoy? We understood. Hybridization korile ba kiho karone korisu to get a desired product, right? As far as the hybridization process is concerned, either you can go for a single cross or you can go for a multiple cross. What is the difference between single cross and multiple cross? If you cross or if you do the experiment between two plants only, that is what is called as a single cross. Kintu tumi ki korisa dudal plant or cross korisa offspring to aru edal plant or logot cross korisa ako tar offspring to aru edal plant or logot cross korisa that is what is called as multiple cross, right? The more you keep on multiplying, the more qualities would be added to the recombinant. Yes or no? Understood? Right? So either we can go for a single cross or we can go for a multiple cross in the process of hybridization. As far as the examples are concerned, not important from examination point of view. ঠিক আছে এইবিলাক মুখস্থ নকৰিবা example কিয় দিছো just to show that multiple cross কৰিও আমি improved variety of wheat or improved variety of rice we have made, right? So কিবা doubts আছে নেকি পইণ্টছ কেইটা লিখি লওঁ তোমাৰ এইখিনি লিখা হ'ল অ'কে কোৱা কোনটো পইণ্ট বুজি পোৱা নাই ছিংগল ক্ৰছ মাল্টিপল ক্ৰছ হাইব্ৰিডাইজেশ্যন মানে কি two different plants right if you cross two different plants কি পাবা generations পাবা right অকল দুডাল প্লান্টে ইনভলভ কৰিছা only two plants are required for the cross if that is the condition that is called as a single cross since we are making a cross আমি artificially কৰিছো ন naturally to কৰা নাই artificially আমি তিনটা চাৰিটা প্লান্টটো to cross কৰিব পাৰো একেলগে দুটা প্লান্টে যে ক্ৰছ কৰিম not necessary দুটা প্লান্ট ক্ৰছ কৰিলে কি হ'ব দুটা প্লান্টৰ characteristics আহিব বহুত কিটা প্লান্ট ক্ৰছ কৰিলে কি হ'ব বহুত কিটা প্লান্টৰ characteristics if you want one plant to have all the characteristics ধৰা তোমাৰ high yield লাগিব disease resistance লাগিব adverse conditions against resistance লাগিব আৰু কিবা কি উখও হ'ব লাগিব হকটো হ'ব লাগিব মিঠাও হ'ব লাগিব সব যদি লাগে you make your cross right but it sounds very simple কিন্তু it is not so easy also right hybridization or one most important drawback offsprings are not exactly what we want right ami cross to koru so that we can get a desired plant kintu the chances of success is very less right
between more than two parents or
parents or plants ekitai bostu no sorry plants I likha I think this is better এখিনি পইণ্টছ বুলি লিখিব পাৰা artificial hybridization is মানে the first product of artificial hybridization right ta mane কৈ আছে তোমাক লিখি লচোন এটো okay Fairchild Mule Fairchild Mule Fairchild Mule spelling to ধৰিব পাৰিছা Okay. Now the points of hybridization. What is important is just the first point. जोटो निके question আহিছে बाकी इटार इटालके question आहा নাই. Hybridization की होय? Cross होय. Hybridization artificial होय ने natural होय. Both the two types of hybridization are seen, right? Natural hybridization होय, artificial होय. Natural hybridization first code report करिसे in maize, right? Artificial hybridization means you are doing a hybridization. The first hybridization was done upon two plants. What are the two plants? Sweet William aru Carnation. Tahator jinto offspring asile. The name of that offspring is Fairchild Mule. So what was the first artificial hybridization experiment or the first offspring? Fairchild Mule. And the mule kole hai donkey mule hetu na bhabiba aru no. That's not. पहले वही सोचा था ना, दो प्लांट्स का कॉम्बिनेशन और म्यूल प्रोड्यूस हो गया. वेरी नाइस. और बाकी फर्स्ट टर्म के बारे में क्या सोचा था? वो तो हाँ म्यूल का नाम है ना. No, it's a cross between Sweet William. It's a flowering plant. Carnation is also a flowering plant. तार जिंटू offspring तार नाम Fairchild Mule. So ultimately what we have learned is mutation, plant breeding could be done by the process of hybridization also. Ekta hol liki. When we talk about mutation breeding, plant breeding, mutation breeding, breeding means technique of cross, right? By the process of what? By the process of mutation. So during this, do I need to write the definition of mutation? Genetics or porisa, what is mutation, right? Mutations could be both natural and artificial, right? Hetu to jano, artificially jodi ami mutation koru, tak ami ki boli kau induced mutation, hekin likhi dibo laibo na jano. It is zanim, no? Definition of mutations, you have to read it only. ইয়াতে নামকেইটা মুখস্থ কৰিব লাগিব হুইট ভেৰাইটি ৰাইচ ভেৰাইটি Some Pusa Lerma, P-U-S-A, Pusa Lerma, L-E-R-M-A. এইকেইটা নাম দুইটা জানিব লাগিব দেই just to have an idea that hybridization has been conducted on different crops. তাৰ ভিতৰত maximum question has been asked about wheat and rice. কেতিয়াবা কিছুমান বুকছত barley দেখা পাব পৰা কিন্তু নিটত নাহে From NEET point of view, it's only wheat and rice that we'll study. So we are talking about the methods of crop improvement. The last one is polyploid breeding. So how can you improve a crop? We can improve a crop by the process of polyploid breeding also. We can improve a crop by hybridization also. We can improve a crop by mutation breeding also, right? So polyploid breeding. এইখিনি হৈছিলে নেকি লিখা Okay, up till now what we have written, crop improvement could be done by a process called as polyploid breeding. What is polyploid breeding? You increase the sets of chromosome, that is what is called as polyploid in a particular cell. One cell, generally we have diploid number of chromosomes. We have heard the two terms that's more commonly available, that is haploid and diploid. If you want to increase the number of or sets of chromosome, that is called as polyploid. How will you do polyploid breeding? There are many methods, there are use of certain chemicals, etc. One of the most important chemical that we have already read is something called as colchicine. Cell cycle and cell division ma colchicine or kotha koisu. Colchicine didle ki hoy? Separate no hoy. Separate no hoy mane? The centromere does not split. If the centromere does not split, then obviously the chromatids will not move to the two opposite poles, and as a result the number of chromosomes will keep on increasing, right. So, colchicine, normally polyploid breeding bully kole, out of these this one is very important, right. How will you induce polyploidy? By application of certain chemicals called as colchicine, right. Next, as far as polyploid is concerned, normally it is of two types, autopolyploid and allopolyploid. Autopolyploid or iman questions aha nai. Allopolyploid or question aise. Now you have to understand a little slight difference between both of them. Autopolyploid baki eku noporiba, just a example to swasun. Tamane ki adult plant ase tar bahut genes ase, okol eta gene increase kori goi asa. Adult plantot bahut genes nathake jano, eta organismot bahut genes nathake jano, right. Out of all the genes that our organism have, what you are increasing is just one allele or one gene, right. It is the same repetition. Autotetraploid, it could also be autotriploid. Autotriploid likhile ki hobo, you are increasing a A, 3 times, auto triploid, auto pentaploid. You are increasing the gene A how many times? 5 times. So, if you are increasing the same genome or the single same genome, that is what is called as auto polyploid. But in case of this, read the sentence. Yate ki korisa? You go for a process called as hybridization. Then after hybridization, you get a product which has both the characteristics of both the parents. Tar jonto chromosomes thakibo, tak ami ki korim? Double korim. So, it will be a mixture of genes together. Eta gene increase korisa, ne mixture of genes increase korisa. Eta gene jedi increase kora, it is autopolyploid. Mixture of genes keneke hol by crossing 2 different plants, right? That is called as allopolyploid. Out of this, colchicine monot rakhiba and examples. Your example kita likhi diyo. And this point is also important. Right? Okay, if you are talking about allopolyploidy, that ki koribo lagibo, duta parent cross koribo lagibo. 2 different plants would be crossed, and that product, 2 examples, triticale or raphanobrassica. Raphanobrassica khunisan nai kobo noaro, but triticale khunibo lage. Offspring of what? Wheat and rye, Triticum aestivum or sickle cereal, right? So, sickle aru Triticum aestivum duita cross kori pelai jonto offspring produce korise, that is what is called as triticale. And if you are talking about Raphanobrassica, it is reddish and brassica is mustard. Duita cross kori pelai you get. Ekhini hol. Okay, so crop improvement by polyploid, what you do in this method is you increase the sets of chromosome. How do you increase the sets of chromosome? By maybe application of certain types of chemicals. The most common thing that we talk about is colchicine. While you are going for increasing the sets of chromosome, increase kiman koriba? Twice x ase, twice x by x ase, no? X or twice x, these two are the most common things that are commonly available, right? Twice x or we increase korisa, kiman increase koriba? Thrice x, 4x, 5 times, 6 times, etc. When you are keeping on increasing, one thing that we need to understand is odd number of increase to hodai ki hoy? Sterile. Even number of increase to hodai ki hoy? Fertile. This part not required for your examination. ঠিক আছে, so এইখিনি part মই সোৱা টিকু কৰা নাই একো, অকল এইটো পইণ্টটো মনত ৰাখিলে হ'ল, ৰাইট? যদি বাই চান্স কুৱেশ্যন আহে 8x পলিপ্লইডৰ কাৰণে 8x ফৰ্ম কৰিলোঁ, 8x ষ্টেৰাইল হ'ব নে ফাৰ্টাইল হ'ব? Odd is sterile, even is fertile, right? And how do you do it? Duta beleg crop loba, cross koriba, the generation is called as F1 generation. F1, so notun duta eta beleg plant lole. For example, if you take a tomato and maybe you take a potato, both of them mixed together, cross korile to kiba eta product form koribo, no? That product ki hobo? Sterile. Hey bostutut chemicals di pelai increase kori pelai ami fertile korabo paro. But as I have already written, no, hybridization chance of success is very less, right? Ami jete eta alu, eta bengena, eta potato or eta tomato so mix kori dio, alu pitika khaboi na lage. Why do you have to go on mashing that potato and the brinjal? Plant dalote di dibo dokhon. Hoy na hoy? So gutei kita mix koribo to dorkar nai no, tumi alu eta aniba, bengena eta aniba, bilahi eta aniba, gutei khini ani you'll go on mashing it down. What's the point of wasting time? You have a plant that will give all the taste. Right? If this is so easy, then tenuka plants produce koribo parilu heten, but we know hybridization rate is very less. Ji monjai take bole cross kori disa. Ki hobo? Not only in case of plants, in case of organisms also. GT cross kori disa. Kukur mekori boli eku kotha nai. Just come and cross it down. A2 question ahe de. Triticale is a cross between triticum and sickle cereal. Turgidium, Turgidium. Huh. Okay. Triticum, triticum. Turgidum. Aru epine sickle cereal, SECALE CEREAL. Again, this part not important. ঠিক আছে, important কি অকল এইখিনি polyploids crop এটাৰ example tritically is a cross between, it's a cross between wheat and rye, right? Scientific name important. Scientific name triticum estivum দিব পাৰে, turgidum দিব পাৰে, most commonly estivum দিয়ে. So, we are talking about the steps of plant breeding. What we said is, first we go for collection, then we go for selection of the parent, and then we go for making a cross within the two desired parents to get the recombinant. So, etialoke ami ki pohilo? We have talked about collection, we have talked about selecting parents and making a cross. What is the product that we have right now is the recombinant. The next step would be selecting and evaluating the recombinants. Already ami plant breeding process to arambho korisu. What we have done, we have collected the germplasm, then we have selected the parents, and then within the two parents we have done everything. Either we went for hybridization, or we went for mutation breeding, or we went for polyploid breeding, etc. etc. Right? Ebilak korar pisot, the desired product is called as a recombinant. What next? We need to observe the recombinant and see whether that is perfect or not. So, what would be the third step? এতিয়ালৈকে আমি কি লিখি আছিলো? Second step. Plant breeding or কেইটা step? 4টা, right? First was collection of germplasm, second selecting of parents, third selecting of or supervising or testing of superior recombinants. Superior recombinants আমি কেনেকৈ পালো? By all these? Yes or no? Sequence to বুজি পাইছা নে নাই? কিয় নাই পোৱা? কোৱা, কোনটো বুজি পোৱা নাই? কোৱা. Plant breeding process to ki hoy, you improve the crop. Keneke kora? Steps ase ketaman? Ki steps ase? First, collection. Second, you select the parents. Parents kita select kori thole hobo ne tahator mazot kiba kibi koribo lagibo. Kiba kibi to koribo lagibo, ki koriba? Cross koriba, right? Or mutation koriba, or polyploid koriba. Right? Hey, kiba kibi korar pisot ki hobo? Offspring eta produce hobo nohoy. Hey, offspring to ki boli kom? Recombinant, right? Hey, recombinant to test koribo lagibo na lage. That is the third step. Etialoke ki pori asilo? Second step. Etia third step. Buzi pala? It's so lengthy, no? Ha? Aru edin poriba, pari jaba. right? Third step. What is the third step? The product that you have obtained from crossing the parents. Tamane ami jundul notun plant bonalu. Hey plant dal ami ki koribo laibo etia test koribo laibo. Hedal bhal ne beya keneke test koriba. One method is you have to keep on growing that plant for several generations. Notun plant dal grow koriso, hey plant dal self pollinate koribole dim aru hetuke ki korim. For several generations you need to keep on growing that plant just to check does the quality remains good or not. Jedi eta generation korile nohobo bahut bar tak grow and regrow koribo laibo. Tetia jodi if everything is all right, then only you can release the plant. So what is the chapter? Strategies for enhancement of food production. Means we have to talk about certain techniques through which we can increase the quantity, the quality of the food. Why do you have to enhance food? Because of the growing population. As far as enhancing food is concerned, what are you going to do? We can go for process like animal husbandry, plant breeding, tissue culture, single cell protein, etc., etc. The first that we have studied is plant breeding. What is plant breeding? It's a technique of changing the trait of a plant. Why do you go for plant breeding? To get an improved variety. How will you go for plant breeding? We have certain steps for that, and the 4 steps are collection, selection of the parents, selection of the recombinants, and then testing, right? As far as collection, what you need to remember? The word germplasm. The next word to be remembered is indigenous and exotic, right? Hey duta term ami monot rakhibo lagibo. Then the next one is selection of parents. Talking about parents, how will you go for selection? Either mass selection, clonal selection, or pure line selection, right? Any process you can go through, right? As far as selection is concerned, that method is supposed to be the oldest method, right? Mass selection boli kowa loge loge, it's only on the phenotypic characters that you see. Early ami je rice khabo lage boli janilo, keneke janilo? Because my father said, father kune kosile? তেওঁৰ ফাদাৰে কৈছিলে একদম অৰিজিন কোনে আৰম্ভ কৰিছে? Was he a scientist? যোনে first discover কৰিছে that this seed could be eaten, হয় নহয়? First এডাল plant ভাল, এইটো খাব পাৰিম. Early prehistoric people, they were not scientists. কি চাই পেলাই? Maybe the taste, right? তাতে মানুহ মৰিছিলেও, কি খাইছিলেতো ঠিক নাই ন? Right? So like prehistoric ki korise, it's only on the phenotypic characters that they have worked upon, and it is the result of their study that we are eating the food now. Maximum of the food already discover hoi goisile. Now what we are doing? Making new varieties, right? Making new varieties, first plant breeding, how do you make it, we have discussed. Then selection we have talked about. After that we make a cross, we go for hybridization or mutation breeding or polyploid breeding, etc. We get the recombinant. What do you do with the recombinant? Selection and evaluation. How will you evaluate? Self pollinate koribole dim. Kak self pollinate koribo diba? Jinto recombinant ami bonalu, right? The newly formed recombinants undergoes what? Self pollination for several generations. After that it has to go কোনবাই to certify কৰিব লাগিব না, this is good. মই ক'লে এইটো নহ'ব, you have a department for that. So ইহঁতে যেতিয়া certify কৰিব that this is good, this is good, best, then it goes for commercialization. So this is all about plant breeding. So today in the class we have completed plant breeding. In the next class we will be talking about green revolution. ঠিক আছে, so heading green revolution কৰি থবা. Thank you.